Okay, this is the last video, hopefully, in preparation for the IB exam. I'm going to continue on option B at this point. I'm going to try and keep this as brief as I can. I'm in class today, and posted online is option B, sections 1 and 2. Just wanted to refresh a few things about section 2, which was proteins. So I'm going to go through each point very briefly. You need to be able to draw the general formula of a 2-amino acid. And remember, that refers to a generic structure where the second carbon, counting from this carboxyl group, this is carbon number 2, and it has a think of this as a variable. There are 20 different amino acids in human beings, and each one is different in this location. So the purpose of the R group is just to let you know that that's a placeholder. This is the acid portion, and this is an amino group, hence the name amino acid, specifically a 2-amino acid. You will see the alpha in front. It's not important what that means for us. Just understand that if you're asked to find or define the general structure, that is it. One of the properties of these things, I'm going to have to pause for a second. Okay, we're back. One of the properties of amino acids, which are one of the 4 biomolecules, is a very interesting property, is that they contain both acidic and basic groups. This amino group actually can act as a weak base, and so remember, bases accept protons. So what you have here is an unusual molecule which has both acid and base properties within it, and that gives it some unusual characteristics. So what you're seeing here is a neutral acid where there are no charges present. It's completely neutral. However, if we change the pH, the environment around this, the pH environment of that this particular molecule is in, usually in a liquid solution, for example, it will behave a little differently. So remember, the human body has a pH between typically 7.2 to 7.4. So some of these amino acids, as you'll see, will be a little different structure. So let's make sure we go through this quickly. We did go through this in class. Remember, low pH means high H+. So think of there being a lot of acids around. So when there's lots of acids around, both ends of these will contain that acidic proton. So this proton here that I'm circling, I'll circle it in red. This proton is an acidic proton, so it would probably come off in a basic solution. This section is a base, so as a base it's more likely to take on an H in a acid solution. So when it does take on that H, it would have a positively charge to it, because we can see here this is hydrogen ion is positively charged. So if we go back down here to my little sequence here, I'm going to make a trend. So, excuse me, at high pH we have a very low concentration of H+, or you could think of this as high OH. So if we think this through, as we go this way, the number of H's increases. So what effect does that have here? Well, first of all, at lower pH we have high H+. That means this is definitely protonated, and this base accepts protons. So what we have down at this end is the positively charged species. So let me go ahead and create a little more room here. That's pretty cramped. So at a lower pH we have the form with the extra proton on the amino group. Remember, that proton carries a positive charge, so all amino acids will behave the same, so we'll use the generic R. Well, this is extra proton with positive charge gives this molecule a net positive charge at low pH. As we move from left to right across this trend, remember, the number of H's decreases, and so one of these will give up its proton first. Well, which one of these is more likely to give up a proton first? A base or an acid? Well, acid's role is to give up protons. The base's role is to accept them. So as we move towards the right, the one that's more likely to lose, remember, like strongest Ka values, is going to be the acid. So as we go this way, we get to some point where that acidic proton comes off, while that proton that was originally added to the amine at the low pH that carried the positive charge remains. And when that acidic proton comes off this carboxylic acid group, we get a negative charge. And if you notice carefully, this compound has both a positive and a negative in it. So overall, it's neutral. This compound has a name. This is called a Zwitter ion. And understand that it is neutral. And because this changes across the pH scale, and in addition, it differs with whatever R group is attached, then we can generate a list, which is found in your data book. Let me bring that up. Let me pause for a second and bring up that data. I'm unable to access that data book right now, so you're going to have to refer to it as you watch this video. Remember, it's posted online at the portal site. So this compound is neutral, and so the pH at which this occurs will depend upon which amino acid is present, specifically which functional group. They are named by this functional group here, this R group. So if you check your data book, there's a chart which lists what's called the PI, or in your book, in your data book, I think it's labeled as pH of isoelectric point. The isoelectric point is the pH at which it is isoelectric, equal electronic charge. So each one will have a different PI, or isoelectric point. So it's important to note that at that point, at that pH, it is a neutral compound. And as we shift to the right, we remove even more H's, and now it comes off of the amino group, returning that portion to a neutral compound. Now it has a net negative charge. Okay, so this is important to note. So if you review the other video, it talks about chromatography and the separation of these amino acids. So please understand, here it's net positive, here it's net negative. And so if you control the pH, you should be able to predict what charge the amino acid will have at a given point. If its pH is the same as the PI, it will be neutral. However, if the pH is below the PI, it will be net positive. If the pH is above the PI, it will be net negative. And then you can put a sample, a mixture of amino acids in something like a DNA gel box, like an agarose gel type thing. Wouldn't be agarose, but it's very similar to it. And you put your samples in the wells, just like always, and those that are positively charged will migrate this way, those that are negative will migrate this way, and it's a size exclusion. Just like DNA gel electrophoresis, which means that the larger ones will move more slowly, the smaller ones will move more quickly. So review that video. That was option B, probably the second one posted, just to familiarize yourself with that process, and also check on the website for a file to photocopy from a test prep booklet that shows some images and things that may help you with separation of protons. So in addition to this chromatography issue, in addition to this electrophoresis issue, you're required to know what the primary, secondary, tertiary, and quaternary structures are. Again, please refer to the previous video. And last, you're required to know the major functions of proteins. They include structure, for example, muscle, things like that. They include enzymes. They are the major component of enzymes. They are also involved in some hormone production. Transport proteins in cell membranes, and sometimes in rare cases as an energy source. So that takes us through proteins.